Yogaha Karmasu Kaushalam.
Hello, participants of the refresher course. I am Ravi Vijayvargia, working at the Department of Biochemistry, MS University of Baroda. I welcome you all to the refresher course. Today, we are going to discuss something which I've been using from 2007 onwards, a system to express protein. Okay, so I think a lot of you must be using different systems to express proteins, including prokaryotic systems and eukaryotic systems. But today we will focus on one system, which is expression of proteins in insect cell baculovirus system. Okay, which is part of eukaryotic expression system. And I started using it in 2007 during my postdoctoral work in the US. And today's topic will also include the research part which we have done using this system. And majority of this work which I'll show you today was done during my postdoctoral research at Massachusetts General Hospital, Harvard Medical School, Boston, 2009 to 2013. And also later on, I'll tell you the current research we are doing in our lab. Okay, so let's get started. My current research area of interest is on Huntington's disease, which is a neurodegenerative disorder. Let me use the pointer. Yeah, okay. So it's a neurodegenerative disorder, you must have heard of Alzheimer's disease, Parkinson's disease. So this is a similar disease of brain, but not as common as Alzheimer's or Parkinson's. It's a genetic disorder which is caused because of a mutation in a gene called Huntington. Okay, the location of the gene in humans is on chromosome 4, short arm of chromosome 4, and it is having a polymorphic area on its 5' end in the exon 1. Okay, so the polymorphism is basically a repeat of CAG. It's a CAG is a codon for glutamine. So there is an expansion of this CAG. So within normal population, you can have a CAG repeat between 6 to 34. That is normal gene. Okay, and in some cases there can be an expansion of this CAG where it can be beyond 34. Greater than 34 means X 35 onwards. So 35 to 39 we call it as a semi-penetrant disease, where some people can get the disease while some will not have the disease. While when the expansion goes beyond 39, that is 40 and above, and it becomes a fully penetrant disease, when everybody having this allele, even one allele. Okay, it's a dominant disorder. So amongst the two allele, even one allele has a repeat size more than 40 and above, they will have a Huntington's disease. So basically, this expansion leads to a production of a protein which has expanded polyglutamine tract in their protein, which is in the N terminal of the protein, and this protein is called a mutant protein. Okay. Uh, the expression of this protein in humans is seen to be there in almost all the tissues or cell types, but the highest expression is seen in brain. Okay, so when the protein gets mutated, okay, the cells which are having the highest expression, they misbehave and there is a cell death. And all of you know that the neuronal cells are terminally differentiated. So once the cells are dead, there is no replacement, and the corresponding functions which these cells, neuronal cells carry, and mainly in the brain also, it's the striatal and cortical neurons, they are affected more. So when they are dead, then corresponding functions, especially the motor functions, the psychological functions, psychiatric disorders, okay, and memory, all these are affected in Huntington's disease. Okay, so if you see here, there is also a population studies telling a inverse correlation between the CAG repeat size and the age of onset of this disease. So you see that as the CAG size, repeat size keep increasing, the age of onset keep decreasing. Okay, so there is a genetic connection and there is a inverse correlation. Also, you see there is a effect of genetic modifiers. That means you can have a inverse correlation, but people with the same CAG repeat also, there can be an lower age of onset, or there can be a higher age of onset. That means apart from the original mutation in the Huntington gene, there are modifiers. The genetic modifier means the other genes can have modification which can increase the age of onset or reduce the age of onset. Okay, so this is also a very interesting phenomena in Huntington's disease. And this is what brain, a postmodern brain looks like. So if you see, this is a normal brain, postmodern brain, and this is a brain from an HD patient. So you can see there is a large area here, we can see, which is totally damaged, which the neuronal cells are dead, and you can see these cells are expressing the normal Huntington with the normal polyglutamine tract, while there is an expansion in the polyglutamine tract in the mutant, and that leads to uh, selective killing of the striatal and the cortical neurons, leading to this particular hollow brain. Okay, so what the question we have to ask is, how this expanded polyglutamine tract affect the Huntington protein. Okay, so these are the different things it can affect potentially. It can affect the protein folding and the structure of the protein overall. It can lead to aggregation of the protein, which is commonly seen in case of Huntington's disease. Thirdly, it can also affect the post-translation modification. So, you know, proteins undergo several post-translation modification, including phosphorylation, sumoylation, glycosylation, methylation. Okay, other proteins also undergo this modification, and these modifications are very, very important for its normal function. Okay, so if there is an altered polyglutamine repeat size, whether it is changing the post-translation modifications in wild type versus mutant proteins or not. Finally, if there are changes in the structure and the post-translation modification, eventually it can alter the function of the protein. Okay, so these are the questions which were there when I started my postdoctoral research at MGH, Harvard Medical School. So to address this question, the main thing was to find out what are changes in the protein when we have a expanded polyglutamine tract. Okay, so to address that, we had to look for different expression systems in which we can express this protein with different polyglutamine tract and see how it alters the function, structure and function. Okay, so we had to explore different expression systems for protein. So all of you must be aware that there are prokaryotic expression systems, which amongst which the E. coli system is the most popular one. Lactobacillus lactis also, and some bacillus species are also used, but they have some limitations which I'll tell you later on. So these are prokaryotic expressions, but then we have the eukaryotic expression systems as well. So you can see I have listed some of them. So yeast is one of the very popular, simple but popular eukaryotic expression system. In yeast, we have Pichia system or Saccharomyces cerevisiae system. Then we have insect cells. Insect cells also can be used as expression system for proteins. And insect cell in combination with baculoviruses is a very powerful system, which we will be discussing in length today. Then we have mammalian systems also, where directly at the highest level of eukaryotic system, mammalian cells can directly be used by plasmid or viral infection for expression of your protein of interest. Then there are in vitro expression systems where small amount of proteins can be generated either using the whole cell lysates from rabbit reticulocyte or wheat germ extract or E. coli extract, but that is not part of discussion today. Today, our lecture will focus mainly on how to use insect cell baculovirus combination for expression of protein. Okay, and why we are not using the simpler system like prokaryotic expression system for expression of this big protein Huntington. So the question here is what we want in the outcome. Okay, so as I told, this Huntington is a very big protein. It's a 350 kilodalton protein, and it has multiple post-translational modification site. Okay, so the limitation of prokaryotic system is it does not carry out so many modification because they lack the enzymes which carry out the modification, and also it's not a very good system for large proteins. Like such a big 350 kilodalton protein is a very large protein, so we cannot use a simpler system like prokaryotic system. So the other option we had, we also, yeah, yeast system is a eukaryotic system, but still it also does not carry out all the modifications required because of the lack of enzymes. So we then choose a system which is capable to produce large proteins like Huntington, and also carry out the modifications, and it is almost modifications are similar to mammalian cells. So we chose this system. So today we'll discuss in detail how this system works. Okay, So what are the advantages of using mammalian or eukaryotic expression system? That we get most of the post-translation modifications in them, which will make the protein highly functional. Also, it will be compatible to humans because it's a eukaryotic system, and that will produce low immunogenicity to humans, so immune reaction will be less. And also, if you are using non-human cell lines like insect cells, they will not be susceptible to human pathogens, so that cross contamination will not be there. And most of these systems are very safe to use. Okay, so these are the advantages of eukaryotic and especially mammalian system, and we will be today discussing one of them, which is the insect cell system. Okay, and all of you know that post translational modification in eukaryotic is very important phenomena, and mostly it occurs in the cytoplasm or endoplasmic reticulum, and these are very important for protein activity and stability. And these are enzyme catalyzed reactions. Okay, so they cannot happen on their own, so they need a specific enzymes to be present to carry out this reaction. So like for example, kinases are required for carrying out phosphorylation. Similarly, other enzymes, specific enzymes are required to carry out the modifications. Okay, and for example, disulfide isomerase is an enzyme to carry out formation of disulfide linkages, which is a very important for proper folding of several proteins and for its function. Also, glycosylation of several proteins are very necessary for having a proper conformation of the protein. Also, localization is determined by glycosylation type of sugar moieties it is added. Also, they will increase the stability of the protein. Okay, so that's why the post-translational modification of the protein is a very important phenomena, and it is a must for several proteins for their normal function. Okay. So now let's come to the baculovirus insect cell system, which I am going to discuss today in length. Okay, so as the name suggests, this is a combination of two things: a baculovirus and insect cell. Okay, so as you know, right now we are undergoing a pandemic. It's hopefully it's at the end of it. Okay, so that is also because of a virus. Okay, and this virus, the SARS-CoV-2 virus, has jumped host from a bat to human host, and it is replicating in humans, and that's why the disease. Okay, here also we use a specific virus, which is baculovirus, which is specifically infecting insect cells only, and hopefully till now there is no report that it can infect humans. Okay, so that way it is a quite safe system. So this is a very powerful system where we have to use viruses, recombinant viruses, where we introduce our gene of interest and then allow these recombinant viruses to infect insect cells. And when these viruses propagate in the insect cells, they produce their own proteins. Along with that, our protein of interest also get produced. Okay, so this is the crux of this system. So mainly we use this cells derived from this fall armyworm, and these are the cells which are derived from this, which are used as a system. Let me tell in detail what are these cells. Okay, so yeah, so baculoviruses are large diverse group of viruses that specifically infect arthropod, that's insects, and are not infectious to other animals. These are the two popular cell lines derived from this insect fall armyworm, which is SF9 and SF21, and the scientific name is Spodoptera frugiperta, so SF. Okay, S and F coming from here. These two cell lines are suitable for host expression of several recombinant proteins using the baculoviruses, and can be grown both as adherent as well as in suspension. Okay, so that is the beauty of this cell line, that you can grow in monolayer also and in suspension also. And this is how you have to produce your protein of interest. So you have to first incorporate your gene of interest into baculoviral genome, and this recombinant genome will be then introduced into insect cells. So they are the host of these baculoviruses. So recombinant protein is produced along with the when the baculovirus propagate in the insect cell. Okay, so that is how you produce your protein of interest. Okay, so this is just a summary slide, but I'll go into detail of the steps which are required to produce proteins. Okay, so also, yeah, some of the most common baculoviruses which are used in this system is called Autographica californica multiple nuclear polyhedrosis virus, ACMNPV. Okay, this is the most common baculovirus used for expression systems. Okay, so when we see for expression system, there are several commercially available systems available for this particular like baculoviruses system. and we cannot go into details of all of them. So today I've chosen one of the most popular and most reliable system which we also use in our lab. So it's called the back-to-back baculovirus insect cell system. Okay, so in the kit, what all things come? They have this vector where we clone our gene of interest, that is P fast back. We have a specialized DH5 alpha, DH not DH5, E. coli strain which is called DH10 back. It's a strain of modified strain of E. coli. I'll tell you the properties of this later on. Also, we need a transfection region which is cell pectin and SF9 or SF21 cells. These are the requirements to begin with. So first is what we have to do. These are the steps. So we have to clone your gene of interest into P fast back donor plasmid of choice. Then we have to transform this recombinant P fast back into this max efficiency DH10 back specialized E. coli component cells, which will be helping us to generate a recombinant backmate. Okay. And third step is to transfer the recombinant backmate into insect cells to generate baculoviruses, recombinant baculoviruses, and eventually we have to generate high titer baculoviruses. So these viruses we use to infect more insect cells to generate high titer baculoviruses, and eventually when we have high titer baculoviruses that are used for production of recombinant protein. Okay, so these are the steps involved in the production of recombinant protein using the back-to-back baculovirus expression system. This is the vector map of PFAS back. So you can see here, this is a multiple cloning site. So you have to clone your gene of interest in this using the restriction enzyme sites available here. And you can see there is a this is a promoter region. So the polyhedrosis PH is a polyhedrosis promoter, which is specific for expression in insect cells. Then we have a SV40 polyadenylation. So this eukaryotic genes, they require a poly A tail at the 3' end. So that is introduced here. Then you have a for selection of this recombinant plasmids. In E. coli system, we have the ampicillin resistance marker, origin of replication. Then very important aspect of this particular system is using the bacterial T7 transposones. Okay, so all of you know that transposones are jumping genes. So we put in this vector, we have a transposon 7 left, the sequence is left and right sequence are put here. So in between the two sequences, we have gentamicin resistance marker, promoter, we clone your gene of interest here, and the polyadenylation site. So whatever sequence is between these two transposon sites, this will be transposed from our vector onto the baculoviral genome. So that how it happens, I'll tell you in the next slides. So these are the features of the baculovirus genome, that is the backmate. We call it backmate. So it's a large plasmid, and since it is having entire genome of baculovirus, it is called backmate. The name of that backmate is BMON14272. It is 136 kilobase long, and this particular backmate is present with specialized cells which are E. coli cells, DH10 back. The back meaning the backmate here. Okay. So what are the features of the backmate? It's a low copy number having a mini F replicon. So we don't, because it's a large size, we don't want many copies of it. So it's a low copy number backmate. There is a kanamycin resistance marker on it, so that we can select for backmate using an antibiotic kanamycin. And very most important part is that within this backmate, there is a segment of gene or a DNA encoding LAGZ alpha. Okay, LAGZ alpha gene, which is coding for enzyme called beta galactosidase, which allows for blue-white selection. Okay, so within this LAGZ alpha gene, what they have put a mini attachment site, TN7 attachment site. Okay, the transposon 7 attachment site has been incorporated within this LAGZ alpha gene in such a way that it does not disrupt the reading frame of LAGZ alpha gene. Okay, so in normal cells, in the normal E. coli DH10 back cell, if you do blue-white screening, this is a functional beta galactosidase producing gene, and you will get a blue colonies when you add IPTG xcal. Okay, so the purpose of this is that when you have a transposition, your gene from P fast back transposes to this attachment site on the TN7 attachment site on the backmate, then this LAGZ alpha gene will be disrupted, and upon blue white screening, then you will get a white colony. Okay, so we'll be discussing in detail again, so don't worry. So here what I just said, within this backmate, we have this Lexi alpha gene, and upon blue-white screening using X-gal IPTG or blue-gal IPTG, we get a blue colonies. Okay, and transposition, how it happens? So transposition requires enzyme which is transposase. So for that, this particular DH10 back cells also have a small helper plasmid in it. So this helper plasmid is called TMON7124, and it encodes for the enzyme transposase. Okay, so transposase will be produced through this helper plasmid, and to maintain this helper plasmid, there is a tetracycline resistance also. Okay, so we maintain the helper plasmid within the DH10 back cell using tetracycline selection. Okay, and this TN7 transposition occurs in presence of this particular backmate. Okay, so now what is the strategy? So this is again a graphical representation of the strategy. So you clone your gene of interest. So your gene of interest is now cloned in pFastback. So this is the red one is your gene. Then you transform this recombinant pFastback plasmid into competent DH10 back E. coli cells. So within the cells, as I already mentioned, there is a backmate present. This is a baculoviral genome, and the helper plasmid present. Within the backmate LAGZ alpha gene, we have a mini attachment site for transposon 7. And when you put this recombinant plasmid in this, because of the presence of the transposase, the entire gene sequence between the TN7R and L, this flanking this will be transposed. Okay, this will be transposed from the PFAS back onto the attachment site. Okay, so this will be transposed here. Okay, it will jump into this. Okay, and what will happen? The gene will be now transferred onto the backmate. Okay, so this will be your new recombinant backmate. Okay, uh, and this can be selected by both antibiotic resistance and blue-white staining. So for selection of this recombinant backmate, you have to put 3 antibiotics in the LB agar plate, one for tetracycline for helper plasmid, kanamycin for maintaining the backmate, and gentamicin, because gentamicin resistance is present in this. So to maintain this gentamicin, 3 antibiotics you have to put, and X-gal IPTG, and you can do blue-white screening. Okay, so this will allow for isolation of the. So you select the white colonies where the gene is disrupted, Lexi alpha gene is disrupted. So they will give the white colonies. So from white colonies, amplification of white colonies, you will have recombinant backmate. So you isolate the backmate using the your normal alkaline lysis method. So you will get the recombinant backmate. You can confirm the presence of your gene of interest by doing a PCR with the gene specific primers using this recombinant backmate, and then you can confirm by running a gel. You can run an agarose gel electrophoresis, and on the gel you can see whether the gene of your interest is present. So these are some representative images of your antibiotic and your X-gal IPTG screening. So from my lab only, these are my own lab results. So you can see this upon selection, you can see the LA agarmycin, X-gal and IPTG, and you can see there are several blue colonies, but along with that, white colonies are also there. So these white colonies are picked up and they are grown, and then they are used for isolation of backmate. So you can see these are several colonies from where backmate is isolated. So you can isolate, and then if you required, you have to do a gene specific primer based PCR to see whether your gene of interest is incorporated in the backmate or not. Okay, so that can be done. Okay, and once that is done, then you are ready for, once the confirmation of recombinant backmate is there, then you are ready for transfecting the cells. So the SF9 insect cells are the most popular cells which are used for transfection. So this is under microscope, they look like round cells. These are the various properties of insect cells. So these are derived from the pupal ovarian tissue of Spodoptera pugiperda, and they grow both in mononuclear and suspension. They can be adaptable to serum-free media. They can be suitable. They are suitable for transfection, generating high-titer viral baculoviral stocks, and expression of recombinant proteins. The cells are regular in size, easy to manipulate. They allow all the post-transcript modification because they have the enzymes required for it. Also, the doubling time of these cells is 72 hours. Okay, so these are the features of SF9 cells. This is the strategy. Once you have the recombinant backmate containing your gene of interest, you have to transfer that into insect cells using the self-actin transfection reagent. And once you transfer the cells, the recombinant backmate enters the cell, and it will produce the recombinant baculoviruses. So the baculoviruses don't like jump like this outside of the cell. This is just to show that once the baculoviruses are produced in the insect cells, they will divide within the cells, and when they divide too much number of cells in the each cell, too many baculoviruses are generated, then they burst open the cell and they come out in the media. They don't jump into the air, but they are present in the media. And you can see within this baculoviruses there is a small red dot that is representing our protein of interest coming from our gene of interest. Okay, so this our protein of interest is also produced in the viruses produced. Okay, now we have to amplify. Okay, this in initial transfection we have only few cells which get transfected, so lower number of this baculoviruses will be produced recombinant. So we keep the media with us, and then this media is utilized to infect more cells. Okay, so we infect more cells using the medium. These cells, more cells will again get infected with viruses. They will have again, the viruses will divide within these cells, will produce more viruses. Then again, once they divide and they burst open the cells, we collect the second P2 viral stock from this, and then use this P2 viral stock to infect more cells. Okay, so like this we have to amplify the titer of the virus, increase the titer of the virus. And once we have the high titer P3 viral stock, we are ready to infect large volume of cells to produce our protein. Okay, so this time when we have large volume of P3 stock, for example, we collected 1 liter of P3 stock. Now we have to produce protein. So this time what we do is we normally take say, because now till now we are doing adherent culture. Okay, but now we have to use suspension culture because we want to use large volume of the cell. So like same as we grow bacteria, we can grow the insect cells in a large volume of in flask by shaking them. And normally these insect cells require 27 degrees to grow. So 27 degrees, and in shaking incubator maintain at 27, we can grow large volume of cells. And normally we infect at the density of 1 million cell per ml. So say suppose we want to infect 1 liter cells, so we'll take 900 ml of cells and add 100 ml of P3 virus in that. Okay, but this time we don't want the cells to burst open. Okay, we want our protein not to come out in the media, so we allow infection for a limited period of time only, say 24 to 48 hours, which we have to for different proteins we have to standardize condition, but normally 24 to 48 hours only. And after that we collect the cells, we can then now suspend in our buffer of choice, lysis buffer of choice, and then we lyse the cell, and then we can go for protein purification. Okay, so how do we know that our cells have really got infection? So we have to use a control where we don't do the transfection or infection, and along with that we have our infected cells. So you can clearly see here, when the cells are not infected, they will grow rapidly grow and double in the 72 hours, while after 72 hours in the infected cells, you can see they have not divided and the cells look much larger, so they are swollen. So this is a good indication of that the cells have got infection with baculoviruses. So once you are confirmed that the cells have infection, that means now you can go for amplification of viral titer. You do P1, P2, P3. After P3, you can now go for large amount of protein production. Okay, so now this is an example which I was quoting. So this is the purification of the protein of my interest, huntingtin. So we purified a range of different polyglutamine tract in them. So these are, you can see that this is a large protein. So this is 250 kilodalton molecular weight marker, and our protein is 350 kilodalton. So it is coming much higher than 250 kilodalton. And you can see that we have purified 5 different protein with different polyglutamine tract. So these two are the wild type ones, and these three are mutant ones. Okay, and you can see the purity. So we have used flag affinity purification. So we have added a flag tag with our gene. So the flag tag allows us for affinity purification, and that helps us to get very pure protein. So you can see there is no contaminating protein. This is very pure protein. We confirm the protein again by Western blotting using the specific antibodies for this protein, and in that also we get a very specific band. So now the question is that initially we raised what all things we can do with this purified protein. Okay, so our question was like how this expanded polyglutamine tract is, whether it's altering the structure of the protein or it's altering the post-translation modification of the protein. So those all questions now we can answer because we have the reagent in our hand. Okay, so then we started asking these questions. So first thing we wanted to predict that, so this is a prediction. So you can see uh, Huntington prediction. So we can see in this all these orange lines, they are alpha helical regions, okay, and the yellow ones are random coil, and the black one are the beta sheet. So you can see that principally majority of the protein is alpha helical, okay. So that is a prediction. Now we want to confirm by doing experiment. Okay, so we subjected our purified proteins uh, to check by doing circular dichroism CD studies. Okay, so you can see this is 223 Huntington, which is a wild type Huntington, and 46, which is a mutant Huntington, and 78 again is a mutant Huntington. So you can see that as predicted, all of them show a pattern of alpha helicity. Okay, so normally alpha helical proteins will have this signature in CD. So they will have a peak at 192 nanometer, and two troughs 208 and 220 nanometer. They will have a trough. Okay, so all of them, they show the similar pattern, but the level of this can be quantified, that how much is the intensity of alpha helicity of this protein. So if you quantify, you can see here that there is a inverse correlation, okay, or not, sorry, direct correlation. So larger the glutamine length, larger is the helicity. Okay, so the mutant protein Q78 67 46 will have higher alpha helicity than compared to the wild type protein. Okay, so this is the first finding, that despite having the expanded polyglutamine tract, they retain the alpha helicity, so they are alpha helical, but the extent of alpha helicity increases in the mutant protein. Okay. So next we wanted to see the three-dimensional 3D structure of the protein. Okay, so secondary structure, we know it is a mainly alpha helical, but what about the tertiary or quaternary or 3D structure of the protein? So for that we have to purify the protein. You can see this is a blue native page on which we run the sucrose density gradient. So you can see on a native page the Huntington protein shows multiple bands because it is forming oligomers. So you can see this is a monomer, and there are higher sizes also. Okay, so for doing the 3D structure study, we have to isolate the monomer part only. So for that, we do a sucrose density gradient, and you can see in the different fractions, different these oligomers come in different fractions, and these 7, 8, and 9 fractions, they have the monomer fraction. Okay, so this is for wild type Huntington. Same thing we do with mutant Huntington. So again here also, these are the monomeric fractions. So these monomers are then subjected to electron microscopy. So we do a single particle transmission electron microscopy, and you can see that these are the different structures captured in the electron microscopy, and you can see these structures are not very similar. So we have to do a normalization. Okay, so we normalize the structure using and again, yeah, we had done this using without any cross linker and using a cross linker. So cross linker makes the structure bit compact, uh, and then we analyze the data of several, like more than 1,000 particles for each of the protein, and then based on that uh, we did a modeling. Okay, so this is a modeling. So what it reveals, that there is a spherical shape and have a central cavity. Okay, so there is a central cavity and overall structure is spherical. Okay, and that is true for both wild type Huntington and the mutant Huntington. Okay, and what another thing is, the cavity is overlying the end terminus. Okay, so end terminus is partly hidden in this. Okay, so this is also, you can see there is a subtle difference in the structure. They are not exactly identical, so there is a change, subtle change, and overall they have a spherical shape and hollow central cavity. Further, we wanted to see what kind of interactions are happening during the folding or the tertiary and quaternary structures. For that, we did a cross-linking mass spectrometry analysis, where the lysines within the proteins are cross-linked, and we want to see the lysines which are uh, in close vicinity of each other. They only will get cross-linked. Okay, so after cross-linking, we subject it to mass spectrometry and see which of the lysines were part of the cross-linking and which are free. Okay, and based on the analysis of data from cross-linking, we could come up with another model. So this is another model where we can see that what are the different cross-linking in different proteins, the wild type protein and the mutant proteins, and you can see that The light ones are the weak linkages, and the dark arrows are the strong linkages. So you can see within the wild type and the mutant proteins, there are different kind of linkages, and that tells that yes. And the opening, you see the overall there are 5 domains. So you can see that the vicinity of the domains, N terminal, C terminal domains also are altered in the wild type and the mutant proteins. Okay, so these were the first of its kind studies, because before our study there were no purified Huntington proteins available with any of the groups working on Huntington disease. So this our report was first report to tell about the 2D and 3D structure of the Huntington protein, and we published this entire study in eLife in 2016, and this year I was the first author of this paper. And so this was the first such study which was made possible because we could express this large protein using the baculovirus insect cell system. And this was just the beginning, because once we had the protein, then we could collaborate with several groups, and we could answer several other questions. So next, after this publication, we wanted to ask the question, what next we can do. So since we have the purified protein, we have information that there is an alteration of structure because of the expanded polyglutamine repeats. So can we use this particular information to specifically target mutant Huntington, because there is no cure of Huntington's disease? So can we use this information to identify a specific target for mutant Huntington? So using uh, the purified proteins, we wanted to screen regions which can be very specifically binding to the mutant protein. So aptamers came to our mind. So aptamers are single-stranded DNA or RNA or sometimes small peptide molecules that can bind to pre-selected targets, including proteins and peptides with high affinity and specificity, similar to antibodies. But the good thing is that we can uh, produce aptamers very easily, okay, and they are very cheap, okay. So based on this, we wanted to select aptamers, DNA or RNA based aptamers, which are very specific to mutant or wild type Huntington. And also, right now, several aptamers are in clinical trials for different diseases. And also, the first modified RNA aptamer based drug, which is called Mucagen, is already in market. It is a FDA approved aptamer, which is used for treatment of macular degeneration. Okay, so several aptamers are in pipeline, and they will be soon used for different diseases. So we wanted to know whether we can get a specific aptamer which can target the mutant Huntington protein. Okay, so we did a screening. So here what we do is we used Alexa Fluor labeled purified wild type, that's Q23, or mutant Huntington Q78, and we use this labeled proteins to select an aptamer library containing 3418 aptamers using a microarray platform. We actually got it done through a company. So we sent the labeled protein to them, and they did a screening part. And then what we wanted to see, that amongst this library of aptamers, where all our protein of interest they bind, okay, and they do washing and the detection, and they provided us. So basically, this is a summary. So the library was 3418 aptamers, and amongst them 610 aptamers bound tightly to either wild type or mutant protein, and amongst them there is a fold increase or decrease with mutant protein. And we got a list, okay, so this is a big list where they show the ones which show higher binding with mutant or the wild type. You can see the here, and the differentially binding aptamers are marked here with asterisks. Okay, so these are the different aptamers which were specifically binding twofold more to the mutant Huntington. Okay, so all these aptamers, then we got generated sequences, and then we checked them again using ELISA method. Okay, so these are the different aptamers. We named them MS1, MS2, like this. So mutant specific one, mutant specific, like this. So we got the sequences, and then we did ELISA. So you can see this ELISA based studies with them, and then amongst all them, we finally selected 4 of them which showed the best results. So you can see MS1, 2, 3, and 4. So mutant specific aptamers. You can see this when we put plates with the different proteins and then allow this to bind. You see that all of them show specifically or significantly higher binding to the mutant proteins. Okay, so we came with 4 different aptamers which were mutant Huntington specific, and then we wanted to see check their effect in different assays. So one of the assays which we do for checking the activity of Huntington is a PRC2 assay. So Huntington protein is known to interact with this polycomb repressive complex PRC2, which is having enzymatic activity which modifies histone H3 at by trimethylation at histone H3 lysine 27. Okay, and it is this particular complex is comprised of 4 proteins comprising of EZH2, SUSI12, RBAP48, and EED. Okay, so it's a multi-protein complex, and it interacts with Huntington. Okay, so we wanted to see whether there is an altered activity, and this is already known that it interacts more with the mutant Huntington and increase the activity more. And this H3K27 trimethylation is a mark of gene silencing. Okay, so whenever this mark is present, that means that gene is going to get silenced. Okay, transcriptional inactivation is caused. Okay, so it was already previously shown that mutant Huntington increase the PRC2 activity significantly more than the wild type, and thereby cause a dysregulation or disruption in the gene expression. Okay, so when we wanted to check whether if we add the aptamers in the assay, whether they are able to prevent this dysregulation or not. So you can see here in we used 1:1 and 1:10 ratio of Huntington and aptamer, and you can see that when aptamer was present, compared to this red bar, you can see there is a significant reduction in the activity. Okay, so that means yeah, the enhanced or increased PRC2 activity of mutant Huntington is significantly reduced and brought back to wild type levels by this aptamers. Okay, so that was a very nice significant finding. And also you can see that the interaction with of Huntington with the PRC2 components EZH2 also is reduced when we add aptamers. Okay, so when we don't have aptamer, there is a tight interaction, and when we add aptamer, the interaction reduces. Okay, and that reflects in the activity also. Also, we wanted to see whether this aptamer can pull down endogenous Huntington or not. So we used biotilated form of MS3 aptamer, and then we did a pull down assay. So you can see that in the cells expressing either the wild type or the mutant Huntington, and this is wild type, and so the pull down of mutant Huntington was significantly more using this aptamer. So again, it shows that even for endogenous protein, this aptamer works nicely. And also we checked the co-localization. So you can see we use this is the FM labeled MS3. So you can see this MS3 as a green dots here, and the Huntington protein is labeled in red. So when we do a merge, you can see that this Huntington protein is significantly associated with the mutant cell line only, not with wild type cell line. Okay, so again, the specificity of so this is the top one is the wild type cells, wild type Huntington expressing cells, and bottom one is the mutant Huntington expressing cells. So you can see this yellow color is that they are co-localization, they are interacting. Okay, and this is the quantification of the same. So you can see that the co-localization is significantly more in the mutant Huntington. So this paper was that this all data was published in a paper in molecular therapy nucleic acids, and again this has been cited several times, and recently more groups have worked on this MS3 aptamer, and they have confirmed our finding, and they have did further studies. So it's a work in progress. Also, several other papers also came out of this work because of the use of the purified proteins which we produce. So this is particular again on the role of different phosphorylation sites in modulating the conformation of the protein. Also, we developed in collaboration with other groups, we developed a very sensitive assay to detect the Huntington protein levels in biospecimens. This was a novel alpha ELISA assay, and we published this paper in eNeuro journal. Okay. And also there were further studies in which the proteins were used to determine even more post-translational modifications, which were identified later on after several studies. And this is a paper where more post-translational modifications were reported, and their significance also. And recently, this paper I am showing here it was a preprint in here, but recently, only 3 days back, this paper was finally accepted in human molecular genetics. So here also you can see that the one of the phosphorylation site of post-translational modification which we had determined using the purified protein. Now we in this paper we are telling about the role it plays. So it is very important for Huntington degradation using the CAMP independent protein kinase A phosphorylation of this site. Okay, so this is another very important finding, and it all started by determining this novel phosphorylation site using the purified protein. Okay, so I think all these publications all were possible because of the production of full length Huntington purified proteins, and it's still, the lab is still working on more aspects of it. So currently in my current lab also we are looking at different phosphorylation sites. So currently we are looking at two important sites, serine 13 and 16, which are phosphorylated by casein kinase 2, and this phosphorylation in these sites have shown to be play very important role in the protection from Huntington disease. Okay, so in the mouse models they have shown that if there is a phosphorylation at this site, the mutant Huntington will behave like a wild type Huntington. Okay, so for this particular reason we wanted to screen molecules which can activate casein kinase 2 or inhibit the phosphatases which dephosphorylate these sites. So we screened for several compounds, and so these are the two. So even so, we have to either inhibit the phosphatase or activate the casein kinase 2 and see what effect they have. So we are using first of all is the HD150Q cell line, which express the exon 1 of Huntington disease cells having the 150 glutamine repeats, and this in this the protein is highly, it aggregates very rapidly. Okay, so you can see here upon induction of this protein, this form, these are this okay, the Huntington in this cells is GFP tagged, so we can see under fluorescent microscopy. So you can see this fluorescently tagged Huntington. Okay, so upon induction the amount of Huntington significantly increases and these large aggregates are formed, and then we wanted to treat with our molecules of interest to see whether, and yeah, this is a time course. Okay, so you can see with different times. So this is a 24-hour induction, 48-hour induction, 72 hours, and 90. So with time you can see the amount of Huntington aggregates keep increasing. Okay, so with time the cells will have more and more aggregates, and then we wanted to see whether we have any molecules which can reduce this aggregate. So we have a potential molecule of KT. So when we treat the cells with KT, you can see compared to the control where the induction of aggregation is there, upon KT treatment there is a significant reduction in these aggregates. Okay, so mostly soluble Huntington is expressed, the aggregates are not formed. Okay, so again here also you can see the compared to the control, the aggregates are there upon KT treatment at 1 and 2 micromolar. There is a significant reduction in the aggregate formation. Also, we can monitor the aggregates by doing a Western blot. So you can see here, this is a soluble protein, Huntington protein, and the aggregates are there on the top of the gel. And when we treat the cells with KT 0.5, 1 and 2 micromolar, the aggregates disappear. Okay, only soluble protein is there. Same is quantified here. So you can see the amount of aggregates increase when there is no KT, and upon addition of KT, the amount of aggregates significantly reduced. Okay, so this is, and this is a loading control GAPDH. Okay. Also, it's very well known that in Huntington's disease, there is a altered, or there is a dysfunction, mitochondrial dysfunction is there. So because mitochondria is dysfunctional, so mitochondria is powerhouse of the cell. So they are very important to produce ATP. So it's very well known that in Huntington disease patient, there is a reduced ATP level, and the ATP level reduction is also proportional to CAG repeat expansion. So we wanted to see whether in our cells also there is a reduced ATP or not. So yes, indeed, upon induction of Huntington protein in HD150Q cells, there is a reduced level of ATP. But when we add KT to our cells, the reduction of ATP levels is significant, it's almost diminished. Okay, so there is no reduction. Okay, so levels of wild type are reached upon addition of kt, so we can totally reverse the reduced atp levels if we treat the cells with kt. And overall, this is the model. So normal, in normal case, uh, there is aggregation pathway which is followed. But if we are able to so this is the condition when the serine 13 16 are not phosphorylated. But if we are able to phosphorylate these sites, then we can have less and less of aggregated protein, more soluble proteins. And also we can inhibit the hd or huntington disease pathogenesis. Okay, so that is our current line of work. Okay, so I think with all this data, my previous work and ongoing work, and the role of Huntington purification and the phosphorylation must be very clear to you. Okay. So again, before I end the lecture, I would like to thank you all for patient listening. And also, if you have any doubt, you can always contact me. And I think I would like to end with acknowledging. So these are the people with whom I worked at MGH, Harvard Medical School in Boston. And that time the funding was from CHDI. Current work which I am doing in my current lab is funded by DST Sir. These are all my students in the lab. So majority of the work which we are currently I showed today is done by Mr. Raju. And there are these are my dissertation as well as PhD students. Yeah, with this I would like to thank you all. Okay, so yeah, as I said, uh, this is insect cell baculovirus system is one of the most important system. And what apart from that you can use is mammalian expression system, the PCI expression system, or E. coli system. But what is important here to notice today is what you want from your protein. Okay, so that is a very important question. So you should know that what is your end result. Okay, what you want from the protein you want to express and purify. So that is a very critical question you have to ask before you start cloning a gene or start a procedure for purification. Okay, so as I initially mentioned to you, the most simple systems are the E. coli based system. Okay, E. coli or prokaryotic system, they are very good system, very well characterized promoters are there. For example, T7 promoter. We have several expression vectors are there, like PET series of vectors. Okay, so they are very robust, very well characterized. Okay, only drawback is there that if you want your protein to have a particular modification, okay, which is very important for its function. Okay, and you know this modification, as I pointed out earlier, they are enzyme catalyzed reaction. Okay, so they will not randomly happen. So if you want such modification in your protein, and if you are aware that your protein does require those modification, then it's better to do some uh, survey. Okay, randomly you cannot just jump, and because somebody has given you E. coli cells, or you have access to a E. coli particular vector, that is not sufficient reason to use that. So you should really focus on what is you are going to do with the protein. Okay, so for example, once you purify protein, there can be different ways in which you want to use it. Okay, so some of the most common ways you would like to use a purified protein include, like, you want to generate antibodies against it. Okay, so antibodies, that time, again, there also you have to decide whether you want to generate antibodies as a for a whole protein, or you want to generate for a smaller peptide. Okay, and also if you want to generate antibodies which are also identifying the phosphorylation site. Okay, all those things come into picture. Okay, so that also will dictate what methods you want to use for purification or expression. Again, another thing is, sometimes you want to use your protein of interest for therapeutic use. For example, recombinant insulin is an example. Okay, recombinant insulin is a very commonly used therapeutic protein. So initially, when the recombinant DNA technology was not there, the insulin was isolated from pancreas of pigs, and it used to be very expensive. But now with recombinant DNA technology, you get insulin produced in picture system, insect cell system also. Okay, but again, because there is a requirement of disulfide linkage in that, so that has to be taken care. Then only it will be a functional insulin. Also, recently, we are undergoing this pandemic. So several companies who could make this SARS-CoV-2 spike protein. Okay, so that spike protein played very important role in making so many diagnostic kits. Okay, so those who could produce this protein in large amount fast, they made a lot of money. Okay, so even in my lab, we are also working on COVID project also, where we are making antibody detection kit. So initially, I'll tell you that we had to make it fast. We had to purchase this purified spike protein from one company. Okay, and incidentally, it was also produced in insect cells. Okay, so it came at very high price. So I was a little bit sad that I had means being expert of production of protein and purifying protein, I am buying this protein. But within like 2 months or 3 months, we had already cloned the gene. We had already cloned the gene, and right now we are already expressing this spike protein in our lab. Okay, so we can definitely do it if you know the tricks of the trade. And so now we don't buy the protein anymore, and also we can endogenously produce the protein. So this is what the skill is about. Okay, so if you know, I know here again why we are producing an insect cell. Okay, so why not E. coli now? So again here also you might know that the spike protein is heavily glycosylated. Okay, so this particular modification is required for its normal function. So if you want to use a functional protein, then you have to use a system where the modifications are done properly. So in this case, that's why in commercially protein also they are producing using insect cell, and some companies are using mammalian cells also. But yeah, this is the reason that the modifications need to be present, and that's why they are using this. Okay, so this is again one is for generation of therapeutics, you need very pure protein. Okay, another area where you would like to have very pure protein is if you want to solve the crystal structure. Okay, if you want to solve the crystal structure of the protein, you have to have very pure protein. If there are impurities, then it will not form a crystal. Okay, so if that is the case, again in that case also your protein should fold correctly, then only your correct crystal structure will be formed. Okay, so that all things you have to look into it, then select the right system for your protein expression. Okay, of late, the if you see X-ray crystallography is been taken over by another technique. Okay, where it has become easier to solve crystal structures or solve the structure of the protein, and that recently got the Nobel Prize also, the cryo-EM. Okay, so there are some incidences or incidents like where the protein structure, because protein might not be very stable, or it is not forming a crystal, then how to solve the structure. So to answer those things, like this new technique of cryo electron microscopy has come into existence, and it is solving a lot of those unsolvable structures, because you can basically freeze your protein into a particular dimension, and then you can solve the structure. Okay, so that is also another reason you want to purify protein. So for therapeutic use, basic research, and also for antibody generation. Okay, so these are the different reasons you would like to purify protein for. Also, again, I am reiterating that selection of system is again very important, uh, based on the end use of the protein, whether it's a prokaryotic system or eukaryotic system. In eukaryotic system, again, the simpler ones which I listed initially are the yeast-based system, the Pichia system, the Saccharomyces system. They are having very good yield and advantages. They are simple system like E. coli. They can be easily grown. They are much cheaper compared to insect cell system or mammalian system, which uh, are quite expensive compared to E. coli system. The yeast system is cheaper and can be easily uh, scaled up. Okay, so several therapeutic proteins are indeed made using the yeast system, although they, like for example, Saccharomyces system do have some disadvantages, that they tend to have hyperglycosylation of proteins. So sometimes the hyperglycosylation of proteins will render the proteins inactive. So that problem is there. But when this problem is not there, then you can have a functional protein and at much cheaper price. Okay, but if you have to have certain modification which yeast is also not doing, then you have either insect cell baccalaureate system or mammalian system. Okay, so mammalian system, as I pointed out, is the highest level of system where most enzymes are present, so all modifications will be available. Okay, and as in one of the slide initially I was saying, mammalian system use mammalian cell lines. So and amongst the mammalian system, the most common cell lines like which are used are for the adherent cell lines. These are COS cell lines, COS cell lines. Okay, and for the suspension cell system, the cell lines are used are called NS0 or NSO myeloma cells. Okay, so these cells are very popular. Other cells you might have heard of in mammalian expression system, which are popular, are HEK293, that is the human embryonic kidney cells, HEK293 cells. Then we have HeLa system. HeLa cells also are very popular system. And these cell lines, as I pointed out earlier, they can be used for expression of recombinant proteins, either by using plasmid based system where you have a plasmid with a promoter, like most common promoter which are used are CMV promoter, SV40 promoter. Okay, and sometimes endogenous promoters like elongation factor 1 alpha promoter. Okay, so these promoters are there. And also you can use, similar to what I just explained in baculovirus insect cell system, there are viruses which infect mammalian system or human cells, like example lentiviral, retrovirus, adenovirus, adeno-associated virus. So okay, so these all viral systems also can be used. So in that case, you have to clone your gene in one of these viral vectors, and then these viral vectors are transduced into these cell lines, and they will produce viruses. Along with that, they will also produce your protein of interest. Some of these viruses, they have tendency to integrate in your genome, so that will produce large long-term expression, but also they have tendency to produce cancers. Okay, also sometimes you do not want large time expression, you want a transient expression and non-integrating expression. For example, adenovirus, they can do a short-term expression and non-integrating expression. So such viruses are also important. For example, recently most of you must have taken the Covishield vaccine, and that Covishield vaccine was based on the DNA vaccine. So DNA vaccine based on adenovirus. So in that case, your spike gene, coronavirus spike gene was cloned in adenoviral vector, and that was injected, and so that it gives a transient expression of a spike protein in your body. Also it produces a big immune response, so that way you can produce a good amount of antibodies against the spike gene, and that is how the vaccine works. The Covishield. Okay, so Covaxin is a different kind of antigen. It's an entire live attenuated virus they use, so you have a different immune response. While the other, the Pfizer or Moderna mRNA-based vaccines, they are all of them eventually produce the spike protein in your body to elicit the immune response, but the way they produce is different. Okay, so uh, this is just I wanted to tell you today. I think I spoke a bit fast, so a lot of things were covered fast. Okay, so but if the things are not clear to you, you feel free to contact me. Okay, the email address will be provided to you. And I am in MS University of Baroda, Department of Biochemistry. So if you come to Baroda sometime, do let me know. You come to the department, you are most welcome. I'll be happy to host you. And with that, I end this lecture. Thank you, everyone.